Assalamualaikum and greetings to Dr. Nurjia, Isa and friends. Let me introduce our group member. First is Amsar Razik Khan bin Azam Khan. Next is Muhammad Danish bin Tazurin. And me, Shasha Amani Imran bin Adwi. For today's presentation, we are going to present our finding on the protein biomolecules project. We have three proteins in this presentation: transmembrane protein, peripheral proteins, and lastly, the enzyme. I will proceed with transmembrane protein. The protein that I discovered is cystic fibrosis transmembrane conductase regulator, or the UniProt code is CFDR human. For the function, the cystic fibrosis transmembrane conductase regulator, or in short CFDR, protein helps to maintain the balance of salt and water on many surfaces in the body, such as the surfaces of the lung. The CFTR protein belongs to a subclass of protein known as ion channel. So an ion channel transport electrical discharged atoms or molecules from inside the cell to the outside of the cell or vice versa. The CFTI ion channel in the lung transport chloride ions from within the cell to outside of the cell. So the chloride ions travel through the CFTR protein created tube center on their way out of the cell. Next is the FASTA format of the CFTR protein. It has the length of 1480 and with the mass of 168,142 Dalton. For the domain of CFTR, as you can see in the diagram, the highlighted area and the green structure with the highlighted pink is the domain. It is at the position from 26 to 25, which is at the chain A of the protein. Next is the structure of the protein. The diagram shows the structure of CFTR. It consists of 5 chains, which is A, B, C, D, and E. For chain A is the forest green color, chain B is the red, chain C is blue, chain D is cyan, and lastly chain E is purple. Next is the supersecondary structure. The diagram shows that the different color to differentiate the secondary structure. So the coil structure is in orange, the helix in yellow, and the strand is in magenta. For the possible mutation, is that cystic fibrosis CF is a mutation in the CFTR gene that can impair its normal synthesis or operation. So a buildup of thick mucus results from a mutation in the CFTR gene that impair or prevent the production of the CFTR protein, which in turn causes recurrent lung infection, pancreatic damage, and difficulties in other organs. A recessive disease is one such as cystic fibrosis. The F508del mutation removes a single amino acid from the CFTR protein. So without this building block, the CFTR protein cannot stay in the correct 3D shape. So then the cell recognizes that the protein is in the right shape and disposes of it. Next is the factor that affect the stability of CFTR. The deletion of phenylalanine at position 508 is crucial due to the position of this residue in the tertiary structure of the protein. It probably lies at the connecting position between the helical subdomain of NBD1 and the second cytoplasmic loop of TMD2. As a result of the mutation, there is thought to be a less stable conformation between TMD2 and NBD1, resulting in a protein that is recognizable by ER chaperones and which is then degraded by the ER quality control system via ubiquitination and potassium-mediated degradation. So the method that can be used to determine the structure of CFTR. The molecular structure of CFTR have been determined by cryo-electron microscopy or cryo-EM. It uses an electron microscope with a cold stage and computer with powerful image processing software to determine the 3D structure of small biological complex. That's all for transmembrane protein. Next, I will pass to Danish for peripheral protein.
Okay, I will continue for the peripheral protein. Okay, this is my protein. The acronym is ACADV underscore human. the very long chain specific dehydrogenase mitochondria. Okay, as for the function of this protein is oxidoreductase is an enzyme that we know that catalyzes the transfer of electron from one molecule to the another molecule. So this very long chain specific protein dehydrogenase that catalyzes the first step of mitochondrial fatty acid beta oxidation, and aerobic process breaking down fatty acid into this protein acetylene CoA and allowing the production of energy from fat. Okay, so this is the FASTA format for the first isoform. That we have 3 isoform in this protein. Okay, this is the second FASTA format, and this is the last FASTA format for this isoform protein. So this is the protein binding site. So for the first binding site that we have here is from F to S, that the position at 214 to 223. For the second is YWIS from 249 to 251, and the third is FEG 461 to 463, TND 464 and 2466, and last is Q at 562 at the position. So this is the structure for this protein, that this protein have only one chains that I color and label this protein with the color cyan color, and they have only one chain A. Okay, for the super secondary structure of this protein, this involve coil that I color for this coil with blue color, and second is helix with red color, and strain yellow color. The beta strain is for with yellow color. Okay, for the possible mutation of this protein is a low level of enzyme called very long chains dehydrogenase caused by mutation in the VL, these protein genes. Long chain fatty acid from meal and lipid stored in the body cannot be broken down and digested if this enzyme is absent. As a result, these fatty acids are not turned into energy, that which can cause lethargy and hypoglycemia, two symptoms that are typically of this illness, and an inborn error of mitochondrial fatty acid beta oxidation, which lead to impair long chain fatty acid beta oxidation. Okay, for the factor that affects stability for this protein is defective or partially deleted this protein underlies deficiency, and inborn error of metabolism that has diverse and potential fatal manifestation due to the crisis in energy production during fasting or period of increased metabolic demand. So the discrete mutation in a structurally uncharacterized C-terminal domain region of this protein cause enzymatic deficiency by an incompletely defined mechanism. Okay, for the method that can be used in to determine the structure is we use X-ray crystallography. So that we know that X-ray this protein is we use the hanging group, the buffer condition, the saturation point when the crystal will appear. So the diffraction data were collected at the aggregation laboratory synchrotron, that for the experiment for this. That structure was solved into this value by molecular replacement using PDB ID TB60 and refined by Phoenix Encode software.
Okay, Assalamualaikum and hello. My name is Hamzah Razikhan bin Azamhan, and I would like to talk about the third type of protein, which is the enzyme. So what I picked, the protein name is angiotensin converting enzyme, as known as the ACE or ACE human. Okay, what is the function of this enzyme? First, the ACE, the angiotensin converted enzyme inhibitors, are medications that help relax the veins and arteries to lower blood pressure. Also, the ACE inhibitors prevent an enzyme in the body from producing angiotensin type 2, substance that narrows blood vessels. This narrowing can cause high blood pressure and forces the heart to work harder. So the angiotensin type 2 also releases hormones that raise blood pressure. Okay, the well-known function of ACE is the conversion of the angiotensin type 1 to type 2 and the degradation of BK, which all play an important role in controlling blood pressure. ACE also acts on other natural substrates including encephalin, neurotensin, and substance P. Besides being involved in blood pressure control, ACE possesses widespread functions, including renal development, male fertility, hematopoiesis, erythropoiesis, myelopoiesis, and immune responses. So this is a FASTA format. The enzyme has multiple sequences, so this is one of them. Okay, this is the protein binding site. There are multiple examples here. For example, in the binding site, the position 231, there's a binding site in the sequence Y, and another example is the 390 position, which is the sequence H. Okay, this is the structure of the angiotensin protein. So there are two chains available, which is the chain A in red and the chain B in the purple. Okay, so this is the secondary structures. There are three types of secondary structure that are available in the structure, which is the first is the coil, which is in the color green. The second one is the helix in the cyan color, and the strand in the pink color. There are numerous possible mutations that can occur, which can lead to diseases. So the mutation in the ACE gene are the most common cause of a severe kidney disorder called renal tubular dysgenesis. The renal tubular dysgenesis is characterized by abnormal kidney development before birth, the inability to produce urine, and severe low blood pressure. Okay, so the other possible disease from the mutation of this gene can lead to ischemic stroke, microvascular complications of diabetes 3, and intracerebral hemorrhage. So the factors that affect the stability of the protein, for example, the position 390 to 394 mutation affects the stability and function of the ACE protein, which can cause to abolish dipeptidyl carbo-C peptidase activity when associated with the 988, the K9992. Another one is the abolish of the N-terminal active sites, leading to impaired ability to degrade the hemorrhagulatory peptide and acetyl SDKP In contrast, it does not affect cleavage of other substrates such as choleocytokinin 5. So the method that can be used to determine the structure is the angiotensin converting enzyme in the renin-angiotensin system. Regulates blood pressure by catalyzing the angiotensin type 1 to the vasoconstrictor angiotensin type 2. In this study, the ACE was purified and characterized from sheep lung. So the ACE was purified from sheep lung using the affinity chromatography method. Then the determination of purity molecular weight of ACE with the SDS page. Okay, that is it for me. Like to pass to the next member. Okay, thank you and assalamualaikum.